for inviting me. Thank you for inviting me to be a part of SNEV 2021. I'm really happy to be here. So today I'm really excited to talk to you about our journal, Nature Methods, focusing on our editorial team, a few tips and tricks when you're preparing a manuscript for submission. We'll discuss what we look for when we are assessing a manuscript. I want to talk a little bit about these new open access initiatives that we have from Nature Portfolio. And finally, I will take a few minutes to talk about the MyServe guidelines for reporting on EV research, which I'm sure you all already know. But first, a little bit about me. Like you heard, I did my PhD on HIV vaccines in immunology from the Pasteur Institute in Paris, followed by 2 years of postdoctoral training in the United States. At the very beginning of the pandemic, I joined the Nature Methods editorial team in New York City. And here I am responsible for handling papers in immunology, stem cell biology, and cell biology. I'm active on Twitter, and I would be very happy to connect with you. I always appreciate a good science meme. So, our journal Nature Methods was established in 2004, and since then it's been focused on publishing high quality methodological advances in biology. We have a broad interdisciplinary audience, and we publish on a wide array of topics. So this is just a non-exhaustive list of a few examples of areas that we are interested in. Because we are committed to curating the best and the most impactful methods, we are extremely selective and therefore have a high rejection rate. We get about 200 papers a month, of which 80% is rejected upon editorial review, 10% is rejected at peer review, and the remaining 10% goes on to be accepted for publication. So I'm sure we all realize that now more than ever, we realize the importance of effectively and clearly communicating scientific information. And this is not only to researchers in our own disciplines, but also across disciplines and even to non-academicians. And this is where the editors come in. At Nature Methods, we have an in-house editorial team of 7 scientific editors with diverse research expertise. We are led by our chief editor, Dr. Alison Doerr. If you send us a paper, you will reach one of us. We've all had extensive experience on the other side of the bench, and so we are aware of the challenges that come with academic research. We also have a journalist on our team, Vivian, who's basically the person behind the great technology features and the author files that are published in each issue of Nature Methods. So as an editorial team, we are scientifically and financially neutral, which allows us to make decisions independent of any of these factors. We maintain close ties with the community to keep ourselves up to date on issues like what is interesting, what is important, what are the hot trends in a particular research field, which areas would benefit from methods development, or even what sort of editorial policies and standards should we be implementing. We are constantly reading new research, and we highlight exciting studies in the form of news articles or research highlights, which are published each month. And I'm sure you've seen them on the Nature Methods website. We often commission reviews or perspectives on topics we think could benefit the whole community. We also use our platform to raise awareness and to educate. For instance, one of our recent editorials highlighted the need for inclusion of diversity in research samples. In another editorial not too long ago, we talk about how and when to engage with your editor. So we are all committed to promoting rigorous and reproducible science. And these are words you'll hear me repeat often through the course of this talk. This is really ensured by the very thorough editorial process that a manuscript undergoes every time it is submitted to us. We read each and every paper that we get from the beginning to the end. And at each step, we discuss with our colleagues who have the most relevant or appropriate research expertise to make a final decision. So let's say you've written a great grant and you've obtained some wonderful data, which you now want to share with everyone. The first step is to select the right journal. Think about who you want to reach. For instance, at Nature Methods, we are focused on methodological advances that further basic biological research. So a paper that's focused on drug discovery or preclinical research is not a great fit for the journal's editorial scope. It's not that it's not important or not interesting, it's just that you will not be reaching your target audience if you send it to us. If you're not sure whether your manuscript fits a journal's scope, I suggest you send us a pre-submission inquiry. It's really simple. It's basically the abstract or the abstract and the cover letter of your manuscript that you can simply email one of us or even submit through our submission system. It's basically a way for us to quickly assess your work and see if it's within our editorial scope. Another question I often get is about the cover letter. What is the cover letter even, and do you really need it? Simply put, the cover letter is your chance to make an elevator pitch for your paper. It is always confidential between you and the editor. This is an opportunity for you to talk to the editor, explain to them about what's so interesting about your study, or put it in context about, in the context of other studies, explain the importance of the findings. You could make suggestions for potential reviewers or, suggest reviewers you'd like to be excluded. Make sure you mention if you've talked to one of us in a meeting or at a conference, or, if you've had any sort of interaction. That said, it is not mandatory to have a cover letter, and we will never assess your manuscript any differently if you don't have one. So now that your paper is submitted, it will be assigned to the editor with the most relevant research experience. So what do we look for when we are assessing a manuscript? At the top of our criteria for editorial review is methodological novelty. And this is for Nature Methods. So we look at if a method is truly novel, or is it a small tweak to an existing method? Will it advance research in the field? We try to gauge whether it's broadly applicable. Can it be generalized, or is it so specialized that it would, only be applied to one organism under a specific condition? Is the method validated, and is the data included in the paper? Does the method have applications in answering biological questions? We'll also look for benchmarking data, which is really important because this is your chance to prove that your method is really better than anything else that exists in the field. Now, all of our published research content is peer-reviewed. And once we have decided to send a paper out for external peer review, we pick reviewers based on their expertise and who can basically evaluate your paper in a fair and unbiased way. Our standard is 3 reviewers per paper, but sometimes we might assign more to make sure that all the subjects in the paper have a decent coverage. We always honor your request to exclude certain people as potential peer reviewers, but of course this request has to be reasonable. I once had a manuscript where the authors had excluded the entire country of Netherlands. So don't do that. The reviewers advise us on the quality of the data, whether the experimental approach and the analysis meet the standards in the field. If there is sufficient detail provided that would enable others in the community to adopt the method. And if at all the method is of interest and relevance to the field. Peer review is a crucial part of the scientific process. However, the final decision is made by the editors, and we always stand by our decision. We often have reviewers with opposing views, and we can always consider both sides of an argument before reaching a decision. It's not about how many pro or con votes you get on your manuscript, but more like what is the substance of each argument made by the reviewers. That's what we look at when deciding on a paper. We often overrule reviewers, both positive and negative, and generally uphold a very high standards for review. We are intolerant to any form of bullying or bias, and if we see this happening, we will step in. So, say your paper's back from peer review. Consider peer review as an opportunity to improve the quality of your paper. When you receive the reviewer comments, I recommend that you at least address the most critical points, especially if they have been emphasized by the editor. When you submit a revised version, make sure you provide a point-by-point response addressing the reviewer comments. This is your chance to talk directly to the reviewer. Use this opportunity to explain how you've improved the paper, justify if there are some comments you felt need not be addressed, and really highlight all the new exciting data that you've added. We almost always send the paper back to the peer reviewers, so be polite, be courteous, and professional. If you think that some reviewer requests are unreasonable, talk to us. As editors, we are always really invested in a paper that we are handling, and we want to champion you and help your paper succeed. So if you think some experiments are too unrealistic, we would like to hear about it. Like I said, we are all scientists and we've all spent time on the other side of the bench. So we do understand that sometimes it's just too difficult to do some experiments, be it for funding issues or time, or just you don't have the reagents for it. So anyway, so sometimes in spite of all this, a paper gets rejected after peer review. And this might happen for several reasons, such as if there are like serious concerns about the technical aspects of the paper, or if the significance or the novelty of the paper is called to question, or if the interpretation of the findings are too ambiguous and flawed. In such cases, if we think that the concerns could be resolved, we would consider an appeal. So when you appeal a decision, make sure you mention if you have additional data that might address all these concerns in the appeal letter. Let us know if you think there were factual errors or misunderstandings on the part of the reviewers or the editors. Also, let us know if you have specific concrete evidence of reviewer bias. Appealing is not the best choice when there are subjective disagreements on novelty or significance. It is never a good idea to attack the reviewers or the editors, to demand an explanation for why we've published similar papers in the past, or just to demand that we change our minds without really providing any sort of evidence to support your claim. I've also noticed that often authors cite, they mention the number of hits on the preprint server, or if their paper, or the number of retweets their paper or preprint has got, or the number of likes on Twitter. I can guarantee you we never take into account these metrics when making a decision. So if your paper is not a good fit for our journal, you can transfer it to another journal within the Nature portfolio. And here I should say that appeals and transfers, what I'm talking about, these two aspects, they also apply to rejection after editorial review, that is before peer review stage. Our decision letters will contain a link at the bottom, which allows you to transfer the paper and the reviews to another journal. Sometimes we also recommend transfer to a specific journal based on our familiarity with that journal's editorial criteria. Sometimes we would consult with our colleagues at a particular journal to see if they might be interested in the paper. And this is optional. You can always opt out of transfer consultations as authors. Alternatively, you can ask your editor to contact the editorial team at another journal to find out if they might be interested in inviting a transfer. When you transfer to another journal, the editorial team there makes an independent evaluation of your paper based on their editorial criteria. Of course, if you want a fresh start, it is always a good idea to just make a new submission at another place. Ultimately, we all want that your paper finds a good home. So here, let me take a couple of minutes to talk about the new initiatives on open access that we at Nature Methods are really excited to be a part of. So Nature Methods is already a transformative journal, which means that we are committed to gradually increasing our open access content with the eventual goal of becoming a fully open access journal. We are also one of 3 Nature research journals, along with Nature Physics and Nature Genetics, that are participating in the guided open access pilot. So like I said, as editors, we want to find the best home for your paper. And with guided open access initiative, this all comes together in one submission. So once you submit a paper to Nature Methods via the guided open access, it is simultaneously evaluated for Nature Methods, Nature Communications, and Communications Biology. Editors from all 3 journals work together to identify the best journal for your manuscript. And if the manuscript meets the editorial criteria for potential publication at even one of these journals, it will be sent out for peer review. And then once the peer review reports are back, the editors once again discuss the manuscript together in light of these reviews and compile a customized recommendation, which is called the editorial assessment report, that basically guides the authors through the reviews and the recommended next steps. It's a really detailed report that kind of, it breaks everything down into what you need to meet the revised criteria for each journal. So if you're interested, there's more information on our website, of course, but there's also some very nice YouTube videos that explain these initiatives. But for us at Nature Methods, open science, so at us at Nature Methods, open science goes way beyond open access. We want the methods and the tools we publish to be used by the community and to be used to further innovate and make new cool tools. And so we advocate transparency, which in turn promotes reproducibility. As we commit to editorial transparency, where we are open about our expectations about revisions or explain our decisions to the authors and provide more information about the editorial processes, we also ask that the authors provide sufficient information about the method, provide access to unique materials, custom code, and make all raw and processed data available. And so this leads me to the last part of my talk, where I want to talk specifically about preparing EV papers. I'm sure you all already know the MySIF 2018 guidelines for minimal information for EV studies that was published by the Board of Directors of the International Society for Extracellular Vesicles. This paper was published in the Journal of Extracellular Vesicles in 2018, and these guidelines have been updated from the 2014 guidelines. And there are amendments on the topics of nomenclature, separation, characterization, and functional analysis of EVs. So they're really intended to provide guidance on reporting EV research and ensuring the validity of the findings. These guidelines also support the EV track knowledge base, which was published in Nature Methods in 2017. On the EV track platform, you can submit your experimental design and get a score for the thoroughness of reporting. These are two great initiatives to facilitate standardization of EV research and improve reproducibility and interpretation through systematic reporting of experimental design and methodology. So as an example, I want to show you this recent systematic review that was published last year in 2020 that compared 206 papers on mesenchymal stem cell derived EVs and looked for compliance with the MySEF 2014 or the 2018 guidelines. So for instance, the MySEF 2018 guidelines recommends that EVs should be characterized by at least 3 positive and 1 negative protein marker and 2 methods of single vesicle analysis. The authors here found that 185 out of 206 papers did not analyze a negative marker. The guidelines also suggest uniform nomenclature based on size criteria for EVs, such as exosomes or small EVs, microvesicles or large EVs. The study here found that only 61% of the papers used the correct nomenclature. The rest either did not analyze for particle size or used the term interchangeably. Reporting like this leads to such a lot of confusion and inconsistencies in the interpretation of data. And it not only affects people that are reading it, but also people that are building their data based on this. Overall, the study found that only 12 out of 206 studies met the 2014 guidelines, and only 4 studies met the 2018 guidelines. So the 2018 paper on minimal standards has this really great quick reference checklist at the end of the paper, which is basically a concise summary of all the requirements for reporting on EVs. As of last year, editors across Nature portfolio have decided to recommend that all the EV papers at revision stage follow these guidelines and submit their experiments to the EV track knowledge base. I heard a few of you mention the guidelines when you were discussing your own work, and that's really encouraging. We hope that ensuring rigor and reproducibility will lead to new and exciting discoveries in the field of EV research. So that's really the end of my talk. Like I said, we are always happy to discuss, always happy to hear about what you're working on. You can reach me by email or the Twitter accounts. We also have a journal-specific Twitter that is run by us. So yeah, I'm happy to take any questions, and thank you for listening.